Good morning, everyone. My name is Madison Dodds, and I'm the Institute Program Specialist for Salk Institute's Education Outreach team. Welcome to the 5th Annual Ellen Potter Research Connections for Teachers Symposium. This year, the event is taking place in a virtual space, which has allowed us to welcome educators from all over the country and even internationally. We are so grateful you have chosen to spend a couple of hours with us this morning. Now, this event will be recorded so that it can be used by other educators and potentially even for your students. So please make sure that your video is turned off to make sure we have a smooth recording and your microphone stays muted. So a little bit more about the Salk Institute where we work. So Salk is not an acronym. The Institute was named after Dr. Jonas Salk, and you may have heard his name recently as he was a key figure in another global public health crisis, and that was the polio epidemic of the 1940s and 50s. Dr. Salk was the first developer of a safe and effective polio vaccination. He then went on to pursue another dream, which was to create a collaborative environment where researchers could explore the basic principles of life. Now, this building that's behind me, and is also shown in the image, is our fabulous courtyard. Dr. Salk worked closely with Lou Kahn, the architect, to design the Institute. If you're interested in architecture, there's a really cool virtual architectural tour on our website that you should check out. And that fountain down the center of the courtyard is the River of Knowledge, I believe is what it's called, and it aligns perfectly with the sunset on the equinoxes. And off in the distance in the horizon is the Pacific Ocean. It's definitely a beautiful and inspiring place to work. And nearly 60 years later, the Salk Institute is a world-renowned basic biological research facility. So a little bit about some of the areas of study for the Institute is neuroscience and neurological disorders, aging and regenerative medicine, immune system biology, including COVID-19 research. Some of our scientists are studying how the immune system responds to infection, metabolism and diabetes, cancer biology. We're one of very few designated cancer centers that's actually not attached to a hospital. And plant biology. So for anyone who's currently teaching environmental science, I'd encourage you to check out the Salk's website about the Harnessing Plants initiative, which is an initiative that uses plants' natural ability to sequester carbon as a potential solution for climate change. But you really can't pigeonhole our scientists really into one box because so many of their focuses overlap. It's hard to identify just exactly who's working on what because it's such a collaborative environment. Our presenters today are really unique and special because they are able to lend their expertise to all of these different areas. They apply their skills in microscopy and image analysis to advance research across the Institute. Now, education outreach might not be a part of Salk's core research mission, but as the quote on the previous slide suggests, Jonas Salk really believed that it's our duty to leave the world a better place for the generations to come. And if it wasn't for two pretty incredible individuals, we probably would not be here today. And they are, sorry, I'm admitting people as well. If my team could go ahead and admit participants, that'd be great. And that would be Dr. Ellen Potter, our namesake for our event. Ellen really took Dr. Salk's quote to heart and turned it into a call to action. She is the founding director of education outreach and the reason why we are a department here today. Later, she was joined by our recently retired former director, Donna Mapston, and together they expanded education outreach's presence across the Institute and in the community. Together, they have made an immeasurable impact on the San Diego science education outreach community, and we're so grateful to have them both joining us today. This event, the Teacher Symposium, had been a long time dream of Donna and Ellen, and I know it's been a dream come true, and it was actually a retirement gift to Ellen to start this event. So Ellen, can you believe it's been 5 years? Oh, you're muted.
I would like to thank everybody who has made taken their time today to wake up early and be with us. And I'm going to actually say another quote that our president, Rusty Gage, said just recently in a statement, which is from Dr. Salk. He said, what is important is that we, number one, learn to live with each other. Number two, try to bring out the best in each other. And so this bespeaks an entirely different philosophy, a different way of life, a different kind of relationship, where the object is not to put down the other, but to raise up the other. And that's exactly what all of you are doing in the front line of the classroom, and then also all of the researchers that are here working so hard. So I'm just grateful to have this opportunity to be with you this morning, and really thank everyone for taking the time to learn and to hopefully excite and bring new things into the classroom. So thank you, everybody.
Thank you, Ellen, for joining us today. So the legacy does live on. The Education Outreach Department is now composed of myself and my colleague Joshua Von Trapp, who runs our community-based programs, and we'll be happy to tell you a little bit more about our programs later. Since Donna's retirement in June, Monica Parkinson, the Director of Public Programs, has joined in and helped us continue education outreach at the Institute. So I just wanted to go over quickly what to expect for today. So this is our event timeline. The theme for today is visualizing science, and I think that'll probably make sense after you see our presenters' presentations today. Both the presentation and the demonstration will be followed by Q&A with the presenters, and then later we're going to have the opportunity to collaborate and explore some cool resources. One of the things I am really excited about conducting this event in a virtual space is the ability to utilize the chat. So while if we were in person, pausing to answer every question for every clarifying question or comment that you have would be challenging. In the virtual space, you're able to interact with our scientists directly throughout the presentation. So today, we're joined by a handful of the Advanced Biophotonics core facility scientists, and they are really excited and eager to answer your questions. I encourage you to use the chat. Please take advantage of this opportunity to connect with our scientists directly. Think about questions that your students might have. We're going to save the chat and put them in a transcription of it available in the teacher resources folder after the event. And with that, I would just like to say again, thank you so much to Kara, Pauline, Sammy, Linjing, and Arlo for being here today. We're really grateful for your passion and your willingness to come in on a Saturday. So thank you so much. Now I would like to turn it to our recently retired director, Donna Maxton, to introduce our faculty presenter. Donna.
Thank you, Madison, and thank you to everybody who is able to participate with us today. Madison, kudos on turning this into a virtual event. I think I speak for Ellen and myself, and I'm sure all the teachers at the end of this. We are so looking forward to this, and thank you for being able to find a way to continue this awesome program. So our speaker today, it is my privilege and pleasure to introduce Uri Manor. Dr. Manor is the director of the Wide Advanced Biophotonics Core Facility here at the Salk Institute and has long been a champion of education outreach and the efforts at the Salk Institute. So we're very happy to have him as our presenter today. His primary focus in the Biophotonics Core Facility is the integration and application of optical and charged particle detection technologies to study problems of critical biological significance. So I'm sure Uri will be talking about this, but as a core facility, the facility works with all of the laboratories at the Salk Institute. So many different types of biological questions that can be addressed and answered, partially through microscopy and microscopy techniques come into the Advanced Biophotonics Core Facility. So there's a lot of different types of problems that they work on there. Uri's current research is focusing on developing artificial intelligence approaches to increasing the breadth and depth of the microscopy techniques that they use. His main biological interests are mitochondria, hearing loss, neurodegeneration, and synaptic plasticity, which if you don't understand what that means, he'll be explaining that in his presentation. Uri before coming to Salk, he did his PhD thesis research work with Pakara Kachar at NIH, or through NIH, and his postdoctoral training with Jennifer Lippincott-Schwartz NIH and Genelia Farms. And this is truly impressive. By the time he had completed his postdoctoral training, he had published 17 peer-reviewed publications, which relied on his image and image analysis skills. So we are extremely pleased to have Dr. Manor as our presentation, and Uri, take it away.
All right, thank you very much, Donna, for the lovely introduction. I also want to thank all of the teachers for being here. My wife is actually a teacher, and I just want to say I know how hard and how much of your heart and souls you put into your work every year, but especially this year. So I just want to acknowledge that and thank you all for your time and for everything that you're doing, for helping us all be better ancestors, as Salk put it. So I'm going to be talking about much of the imaging that we do in our core as basically the central imaging center of the Salk Institute. And I just want to point out real quick that this is obviously not, this is a huge team effort, and many of those members, as you saw before, are here today to answer questions in the chat. Sammy, Weiser, Novak, Pauline Wales, Linjing Fang, Tara Shiaban. But we do have other members who are not able to be with us today. So it's a really big team. It's an amazing team. It's probably one of the best teams in the world, if not the best. So I'm extremely grateful to have such an amazing team working with us. So I thought instead of talking about all of the microscopes and the imaging and how it works, it might be more instructive to talk about a case study. Why do we do imaging? Why would you ever want to put something under a microscope? And the reason is because if you can see how things are organized, how they're shaped, how they change over time or in different contexts, you can actually understand how they work. And as you all know, if you want to fix something, you need to know how it works. So in the context of biology, that means maybe we could even cure a disease. And one disease I'm particularly interested in is hearing loss. So I'm going to do a case study on how we can use our eyes to understand our ears. And this is personal to me because I'm actually hearing impaired myself. And just to highlight how important hearing is, I hope you all appreciate. I mean, I know you have students who don't listen sometimes, but maybe, you know, even if they couldn't even hear, it would be even worse. So Helen Keller, who was famously blind and deaf, actually said that if she could choose one sense, either hearing or vision, she would choose hearing. And the reason is because she says blindness separates us from things, but deafness separates us from people. And again, as all teachers know, we are social animals. We learn from each other. We need to interact with each other to really grow and develop and survive as human beings. And one way I like to illustrate it is a little bit dark, is one of the worst punishments you can impose on someone is to put them in jail, which is essentially isolation. And of course, you know, if you want to really punish someone while they're already in jail, what do? You put them in an isolation. So that just highlights the profundity and the impact of isolation, which is what deafness does to you. So we have a vested interest in understanding and treating hearing loss. So before I go into it further, you need to understand how hearing actually works. So you all recognize the ear over here. And then, of course, you recognize, most of you have been to a doctor's office, would recognize that there's this thing called the cochlea inside the ear that's famously shaped like a spiral. So this video is going to show you how that works. So, of course, sound goes into the ear through the ear canal, and then those air vibrations vibrate the so-called eardrum, which then vibrates the middle ear bones, when that third middle ear bone over here then causes vibrations inside the cochlea. Now, if you unwrap the cochlea and look at it as a line instead of a cochlea, what we discovered is that it's actually graded and stiff. So it's actually looser on one end and stiffer on the other end, so that different frequencies vibrate at different regions of the cochlea. And this is a representation of what's happening in our ear right now. So this is one layer of organization in a biological tissue that gives us our frequency selectivity. Now, if you take a cross-section of that long floppy thing that you saw, this is what it looks like. As it moves up and down, it causes these little hairs to move back and forth. And when they move back and forth, that actually stretches open a mechanosensitive channel that allows charged particles, ions, to move into the cell. And that's what sends an electrical signal to the brain which you can see represented here on the left. It becomes positive when it moves back and forth, and you can see that little pulse, and that's a nerve that actually goes all the way back to the brain. So that's how this works. And the reason they vibrate back and forth, I'm going to come back to this, is because there's this structure on top that actually pushes these things back and forth as it moves up and down. So here comes our first microscope image. This is an image acquired by our lead electron microscopy specialist, Leo Andrade. So you can see here in the animation that there are 3 cells here moving back and forth and 1 cell here. And that's represented here. So you have these 3 rows of cells in green and then 1 row of cells in yellow. So each one of these here is 1 cell. And each cell has many hairs. They're called stereocilia that stick out of them. And those hairs are what are actually vibrating back and forth at the frequency of sound, many thousands of times per second. And you can see they have this staircase shape where you have shorter rows and then a middle row and then a taller row and then a tallest row. And that's very important for understanding how this works. But in any case, this is what we call a scanning electron microscope image, and Sammy will talk a little bit more about that during his demo. Now, if you look across the tissue, I mentioned before that it's very stiff and tight on one end of the cochlea and really loose on the other end of the cochlea, and that gives us frequency selectivity. Well, it turns out there's another layer of organization on top of that to help us distinguish different frequencies, which is what we need to be able to make sense out of sound. So on one end, what we see using imaging again is that the hairs are much longer, and this is in the low frequency region. And on the other end, the hairs are much shorter, and that's in the high frequency region. So if you've ever opened up a grand piano and looked at the strings in the piano, you'll see that in the low frequency, the low pitch sound, the strings are really long. And if you look on the other end, they're really short. So our ear is organized in the same way. These little hairs are tuned to the frequency of sound that they're supposed to detect. It's pretty amazing. And again, I just wanted to show a higher magnification scanning electron microscope image to show us these little tip links that I mentioned before. So the shorter rows are connected to the taller rows by this little filament. And if you look really closely, you can see them here, connecting the shortest hairs to the taller hairs. And that's necessary for whenever they move back and forth to cause these channels to open. It's basically like flicking on and off a light switch at the frequency of sound. And that's the signal that needs to get to the brain. So here's a different imaging approach called transmission electron microscopy, where we actually section the tissue into 70 nanometer sections. And Sammy will talk a little bit more about that again. But using that approach, we can get much higher resolution, even atomic level resolution. So using that approach, we decided to look at these hairs, and what you can see is this filament, again, that I mentioned before. And what this is 2 different cells. And in one, the cell is stimulated, it's on. And what that means is that string is really stretched tight, and it's stretching open the channels in this shorter hair here. Whereas in this one, it's really relaxed, and nothing's stretched, nothing's open, there's no signal. So here you can imagine is when sound is happening, and here when it's not happening. And I just want to point out that if this shorter hair, for some reason, decided to grow longer and become taller, then this would become relaxed. On the other hand, if this hair here decided to grow shorter, this would suddenly become tight. And the point is that somehow biology has developed a method to tightly regulate this with nanometer precision so that this works. And you can imagine the point is that if they become too long or too short, the whole system falls apart. So this is really remarkable organization, which again, you can only see with imaging. Another thing we can see with imaging is what happens in certain disease states. So there are 3 genes of many genes shown here that when mutated causes hearing loss in humans and in mice. And one of them is called EPS8, another is called MYO15A, another one's called Whirlin. The names don't really matter. But what's important here is that these are 3 different genes, and if you mutate any of them, then we see something similar happen to all of the hairs. What you can see here is that compared to the normal hairs, they're very short. And we know that all of them are very deft as well. So again, something that we could only tell with imaging is why are they deft? We wouldn't know unless we could actually put this under a microscope and look at what's going on with the hairs. And now we know they're too short. And we believe that the reason they're deaf is because they're so short, they can't touch that overlying so-called tectorial membrane. So one way to measure that is, again, with imaging. So you can see the tallest hairs from each cell actually insert into, this is the tectorial membrane, and they form these little imprints where they insert in that overlying structure. So in a normal person or animal, we can see that. We can see the holes where they insert. And what we did was we looked at deaf animals, and we saw that they don't have those holes anymore, supporting this hypothesis that the problem is that these hairs are too short to actually reach the tectorial membrane, or at least one part of the problem. So to address this, we're using something called gene therapy, where we basically hijack viruses, much like coronavirus, but not exactly. And instead of delivering pathological replication to the cells, we've hijacked them and we've engineered them so that instead they deliver a healthy copy of the gene that is missing in these deaf animals. And here you can see an example of what happens when we do that. So we've engineered this virus to infect those sensory hair cells in the ear. And what you can see is that the infected cells, the hairs are growing back. They're longer than the neighboring non-transfected cells, and this is amazing. So the next question is, do these hairs actually contact the tectorial membrane? Can these animals hear? If yes, then we have a chance to take this to the clinic and actually start treating hearing impaired people like myself, for example. Sammy will talk a little bit about some other research we're doing where we use imaging to actually image something called the mitochondria, which are the powerhouse of the cell, as you all know. And one thing we're interested in is what happens during age-related hearing loss or noise-induced hearing loss, which we believe have similar pathways. And what we found is that before noise, in normal conditions, the mitochondria are all over the cell. So these blue things are mitochondria, the yellow is the nucleus. Whereas after noise, all the mitochondria start to move towards the nucleus. So we're really interested in this phenomenon and what causes that and what happens next. Of course, we don't only image the ear, as Donna mentioned, and as you all know, we're a biophotonics core, so we're imaging for everyone at the Salk. And the Salk is amazing. We have plant, we have cancer, we have metabolic research. So this is just a nice little highlight reel of different structures that we're imaging in the core that are not just the ear. And I'm going to kind of skip through. So we've got some blood vessels from the brain that we can image, and we can actually map out all the branches of each blood vessel in the brain to try to understand how that changes during disease. This was acquired during the Zika crisis, and we knew that the Zika virus actually remodels the branching of the vasculature in the brain. So this is a cool example of how we can use imaging to understand what's actually going on. We can also image the neurons in the cortex, so that's what this is over here, color-coded for depth. So the closer it is to you, the more blue it is, the further away from you it is, the more red it is. It's a way to visualize the three-dimensional structure of brain in this case. And we can also image live animals in this case. This is a live worm that's being treated with a stimulus that sort of mimics a predator. And we can see which neurons turn on and off when a predator is present versus absent. So we can actually start to map the circuitry of these responses. And then we can actually start testing drugs to see whether they modulate that circuitry. Now, if we want to really see what's happening in the circuitry, we need to use something called electron microscopy. And then we can start looking at each synapse in the brain. So that's what we're looking at over here. This image was taken by Sammy, Weiser, Novak. And here's the so-called presynapse, and here's the so-called postsynapse. And in the presynapse, you can see these circles. Those are presynaptic vesicles. And they carry neurotransmitters that when this cell becomes excited, let's say by sound, that's going to release vesicles that then that merge with the membrane here and release the neurotransmitter that crosses the synapse, and then that activates the postsynapse. And that is the basis of the circuit, basis of learning, and the basis of memory. So we're really interested in imaging these structures on a whole brain level. Now, I'm going to switch gears, sort of, to talk about artificial intelligence. Before, that was an image of natural intelligence. So now I'm going to show you a little bit about artificial intelligence. So in my other life, I'm a photographer, and I read photography blogs, and I came across this article that showed how you can use artificial intelligence to increase the sharpness and detail in photos. And I realized that maybe we could do this for our microscope images. Now, artificial intelligence, and in particular deep learning, has been around for a while. It's most famously been used for something called segmentation, which is where you take an image and you identify different objects. And the most famous example is self-driving cars. So on the left is what we call the input image. It's just a camera view of a street scene. And on the right is the output image that was created by human beings, where they label different structures in that scene. For example, the street. Car needs to know what that is. Sidewalk, cars, houses, trees. So the way artificial intelligence and deep learning works is you have input and output images. Output is so-called ground truth. And you teach this neural network, starts to learn by looking at many, many pairs of input and output, starts to learn the relationship between them, and it can start to be able to predict the output from any input, even if it hadn't seen it before. But this idea of using deep learning to create a sharper output from a blurrier input was newer to me. But I thought it would be useful for our microscopes, and I'll explain why here. So if you look on the left, we're acquiring an image at low resolution, and on the right at high resolution. These little circles you hear are those presynaptic vesicles I was telling you about. Now, we would love to be able to image all of these vesicles in the entire brain. But if we image at the resolution on the right, it will take 160 years to image one mouse brain. On the left, it'll take 10 years. But of course, on the left, we can't see those vesicles. So this is a really nice example of the trade-off between imaging at low versus high resolution. So you can imagine that if we could image at the speed on the left, but get the results on the right, a lot of people would be really happy. So that's what we've done, is we trained a deep learning model to take these low resolution images, those blurry, noisy images on the left, and convert them into the image that you see in the middle, where now we can see those nice vesicles. And on the right, we took one reference image to show whether those vesicles are real or not. And it's not just making stuff up. And the answer is they are real. So we're very happy with that. And of course, we can also use it for live fluorescence imaging. So on the left is the live image of mitochondria and a cancer cell. And they're actually moving around the cell and merging and dividing all the time. But if you want to see that, you have to image with really high resolution. But the problem is then using a high-powered laser and you're cooking the cells. And now you're studying culinary science instead of biology. So instead, we decided we would image at very low resolution, low laser power, and you get the image on the left, which is pretty noisy and difficult to understand. And then we run it through our deep learning filter, and you get the image on the right, which we can now understand. So that's very exciting. So I want to talk about one other artificial intelligence example of image analysis that we're doing at Salk that I think you'll all find exciting and interesting. So you heard mention of the Harnessing Plants initiative at Salk, where we're trying to figure out a way to make plants more efficient or to make more efficient plants to capture more carbon out of the atmosphere to fight climate change. As it turns out, one of the best ways that plants capture carbon is in their roots. Their roots are very carbon rich. So to understand, to capture more carbon, we would love to have more elaborate and better and more efficient root systems. Of course, roots are also important for surviving in a climate change world. If you want to deal with, drought or excessive heat, roots are very important. So we have a vested interest in understanding how roots are organized. So to do this, in collaboration with the BUSCH lab, we developed this robot that can image thousands and thousands of root systems from many different plants from all over the world or from different genetic variations. So now we can get images of these different roots and start to understand how plants make better or worse roots. But to understand it really, we need to convert these photos into diagrams like this, where we can actually measure the length of the roots and the properties of the roots. Now, Wolfgang has developed a method using computers that can convert these images into numbers, and it's called BRAT, which is a wonderful name. The problem is that it doesn't work so well once you start dealing with more complicated roots, which are the roots that we want to study. And I mentioned before, artificial intelligence has been really successful for being able to interpret images, for example, in the case of the self-driving car. So we literally took almost the exact same algorithm that was used for self-driving cars and applied it to these images of roots to see if we could train it to detect everything we need to detect. So here's an example. On the left is the BRAT result, and on the right is the artificial intelligence result. And every time you see a red dot here, that's a successful analysis. So you can see it's much better than the previous generation. So it's just a great example of the success of artificial intelligence. So here's another example. Here's the raw photo from our robot. And here's the ground truth, which importantly was generated by humans. Humans looking at the image. We've got amazing brains, amazing eyes. So we can look and we can say, yes, that's definitely a root over here. And we can draw with a mouse or on a touch screen where the roots are. And then on the right, you can see the prediction about artificial intelligence, which you can see is very good. But to get better, we need more data. So we developed a citizen science crowdsourcing site on ZOONIVERSE, where people, high school students, for example, can look at these images on the computer, and they can help trace these images, which gives us data that we then feed to our artificial intelligence engine to get better and better at this. And as we get more and more data, we're going to need more and more help. And eventually we'll have a super powerful AI, artificial intelligence, that can work for all of the images we ever get again. So I invite you all to help us out with this effort and spread the word. And check out the link over here where you'll be able to find all the instructions on how to do it and how to contribute. And with that, I just want to thank a really long list of supporters and collaborators, and thank you all for your attention. And I'll take any questions you have. Thank you.
That was wonderful. Thank you so much. For the teachers, we do have a link to the crowdsourcing website that Uri was referencing, so you will have access to that later. If you would like to unmute yourself, you're welcome to use the hand raise feature, or you can type your questions into the chat. And while teachers are typing, I would be happy to kick it off. Can you talk a little bit about maybe something teachers did to nurture your love of science, or on the flip side, something that they did that discouraged you at some point?
Me? Okay. Well, I mean, throughout my life, teachers have always been so inspiring and supporting. As I mentioned, I am hearing impaired. And I had a much more difficult time hearing when I was younger than I do now. Even though my hearing is technically getting worse, my brain is getting better at filling in the gaps of what I don't hear. But when I was younger, I really suffered a lot. And I had a whole team of teachers who worked with me and, really helped me stick with it. And all the way up to grad school, I had teachers helping me believe in myself when I didn't. And I'll never forget that. And in terms of teachers not helping, I think the only thing I can say specifically was I didn't like biology, ironically, when I was a high school student. And it was, I think, a large part because of the textbook, but there was so much memorization that I didn't know why I had to memorize every molecule in the Krebs cycle or all of these things that we were being tested on that really killed. I never understood just how interesting and powerful it really was. So that was something that I had to recover from. Obviously I have, because now I'm doing biology, but it took a long time for me to recognize the beauty of biology. So if I had to choose one, that would be it. I see a ton of questions in the chat, but I don't know where to start. It looks like people are doing a really good job of responding. How long has it taken me to reach this point in my research? Well, I'm 41 years old, so that's one answer. But let's see, I graduated high school when I was 17, and then it took me some exploration. I was a bartender for a while. Dropped out of college, spent 3 or 4 years doing damaging things to myself, and then realized I didn't want to be a waiter or working in the restaurant business for the rest of my life, and I wanted to do something with my brain. So I went back to college, and when I went back, I was so excited to learn. I felt so grateful to have access to all this knowledge and this information. And I think it was good for me to take that time off to mature and really recognize what a gift it is to be able to learn this advanced knowledge. And then from then on, it was a straight path to where I am now, straight from college to grad school to postdoc here at the Salk.
Can I ask a quick question about all the collaborations you have at the Salk and how you came to the Salk and why you feel it's such, at least I feel it's a really unique place to be. So
it really is. It's an amazing place. And anytime I ever go visit some other place, to give a seminar or whatever, I'm always so glad to come back to the Salk. It's truly magical. And one thing, Madison mentioned in the beginning, there's a really unique architecture at Salk, and I never really appreciated or believed how much architecture affects your mind, your emotions, and your spirit, and in this case, an institute, until I was at the Salk. So the amazing thing about the Salk that I think no other place has is, ironically, how small it is. It is really small. There are fewer labs at Salk than some of these big name universities like Harvard or whatever have in its single department. So there might be departments at other universities that have more labs at Salk, all studying one thing. On the flip side of the Salk, what we have is a combination of small but powerful, or really high caliber. So we might only have a small handful of plant biologists, but they're the best in the world. We might have only a small handful of neuroscientists, but they're the best in the world. And they're all interacting because the place is so small. So you might get a neuroscientist talking to a plant biologist about branching. I showed you that image of blood vessels in the brain branching. That's the same thing that roots do. And it turns out some of the same mathematical principles work for both of them. And there are tips and tricks and tools that were developed by plant biologists that neuroscientists would never have been aware of, but they can use them, and vice versa. So these common principles in biology, in nature, in mathematics can now be leveraged by having this nice interaction in this small place between all of these world-class scientists. And there's no other place in the world I've been to that does anything close to that, where you have the breadth and the depth. So as a core director who collaborates with all of these people, for me, it's just like, it's almost too much. It's like overstimulation of amazingness. I get to see all of it and work with all of it. I wouldn't trade it for anything.
thanks, Uri. You did a great job on how we all feel about the Salk. So I appreciate it.
Sure, thank you.
I have another quick question. Is there any new technology or any other tools that we don't have at Salk that you would like to see us acquire?
That's a really bad question. Because of course there are. I'm a technophile. I've explained to people, my hearing aids, I wouldn't be where I was if it wasn't for hearing aids, right? So I have a direct personal connection to how tools really make life a better place and allow us to do things that we couldn't do before. And of course, there's always, just like computers, every year there's a newer and better version coming out. And, when I came here, I bought a bunch of new microscopes. At the time, they were cutting edge. That was 5 years ago now. So there are new cutting edge microscopes that I would love to have at Salk. The problem, of course, is that they're expensive. Like one microscope is $1 million, or between $1 million and $10 million. So that's why we don't have them yet. But yeah, there's new kinds of electron microscopes that will give us better resolution to be able to resolve things like synapses a little bit better, or mitochondria. There are certain fluorescence microscopes for live imaging that are faster and higher resolution that we don't have yet that I would love to have. Those are the two key things. And a couple other smaller toys that would be nice to have as well. So if anyone's playing the lottery, you know, keep us in mind. Someone just asked if I collaborate with other researchers out of Salk. Yes, many. The majority, you know, outside of Salk, most of our collaborators are at UCSD just by virtue of proximity. It's easy for them to bring us samples. But we also collaborate with people at Genelia Farms, in Berlin, in Israel, in China, in Japan, and the things like artificial intelligence are a great place for collaboration because all you need is the data. So we can send them computer code via email. We can send data via file transfer online. So there's really nice ways now that we can collaborate with people all over the world, and we do.
All right, is there any final questions? Oh. Yeah, we can answer both.
So 2 questions. So are other labs using viral vectors to help with hearing? Yes, we're definitely piggybacking on a lot of hard work done by other labs. Most famously at Harvard, there's a couple key labs there that have been working hard to develop these virus methods that are much more efficiently targeting the inner ears. But of course, as I showed you in that one slide, there's 3 different genes that cause the same thing to happen. There are even more genes than that. So no one else is working on the specific gene that we're working on, but other people are working on similar methods for other genes. Someone asked if there are a library of these images available to teachers. First of all, we're happy to make available any and all of the images we showed and more, if anyone wants to use those. Also, across the street at UCSD, there's something called the National Center for Microscopy Research, NCMR, NCMIR, and they have something called the Cell Image Library, which is really worth checking out. It's a collection of many beautiful high-resolution images of many different types of cells and structures in cells. Great. Okay.
Thank you so much for that fantastic presentation. I know I definitely learned I think the idea that we can visualize hearing is an interesting concept, and I think it is really important. So thank you so much for sharing your passion with us this morning. So now we're going to do a little bit of a transition, and we are going to go to our demonstration portion, which is going to be put on by another member of the Biophotonics core team. So Sammy Weiser Novak completed his bachelor's degree in biomedical toxicology in Ontario, Canada. Sammy moved to the University of Victoria to perform his master's in the laboratory of Patrick Nearherini. Sorry if I butchered that. The author of Netter's Histology. Sammy uses light microscopy and electron microscopes to study the relationship between structure and function in living things at different spatial scales. As a core facility microscopy specialist, he can help other researchers with experimental techniques that could take years and millions of dollars to develop independently. His interest in science peaked in grade 7 when his teacher showed him how to look at swamp water in his own blood under a light microscope. So he knows firsthand what a difference teachers can make. Please welcome to our virtual symposium, Sammy.
Hey, thanks so much for that wonderful introduction, Madison, and thank you all for being here today. And thanks, everybody, for that great talk. I think it really set the stage for this demonstration here, hopefully. So I'll go ahead and share my screen here. And hopefully there's plenty of continuity between Uri's presentation, mine, and I'll try to give a little bit more of maybe a technical kind of overview over how we generate some of this data and kind of give you a little bit of insight into the nitty-gritty. So briefly today I'll talk going from tissues to nanoscopic models and insights, nanoscopic being at the nanometer scale, and I'll be demonstrating some of this idea of getting a 3D electron microscopy data. So we'll start with some, warm up with some deep thoughts, and then go on to kind of remember what resolution and means and why we bother with electron microscopy when it's expensive and takes a lot of time. And then kind of go into the technical workflow briefly and go into some of the applications that, and I'll highlight some of the work that I've had the chance to work on with collaborators, and also some of the work of my colleagues that are on the chat now at the core. And hopefully this will provide kind of a technical overview of what 3D electron microscopy can be used for, inspire a lot of maybe more questions than answers maybe, and ideas for lesson plans and stuff, and hopefully we'll be able to work some of that out in the question time afterwards. So, deep thoughts really quickly, what is life? There's an interesting paper of 2012, this idea of self-reproduction with variations. There's also this idea that inside has to be different than the outside, and that takes energy. But generally, the point is that these phenomena, how we keep the inside different to the outside, how we replicate, it all tends to happen at a very small spatial scale. Where a human hair is maybe 0.1 millimeters or 100 microns, a blood cell might be 20 times smaller than that, and a nucleus might be smaller than that, and then the other kind of constituents of cells might even be smaller than that. And the synapses that Uri was showing are much smaller, maybe 100 times smaller than a nucleus, 1,000 times smaller than a nucleus. And we'll kind of get to that in a second. Really quickly thinking about what resolution means, kind of looking up instead of down in the astronomy world, there's an idea of binary stars, and as we're looking through the atmosphere and we see twinkling stars, that's actually the water kind of getting in between our eyes and the star, and it kind of creates aberration as the light travels towards our eyes, it gets scattered, and so we see twinkling. But if we want to resolve whether a star is a single star or a binary star or a galaxy or some other phenomenon, we can correct for the aberration in the atmosphere, and so we can increase our resolution. So the resolution, the idea you can see the two spots instead of just seeing one blob. And the idea of resolving power in microscopes. So how does resolution work in microscopes? We're not just magnifying stuff, we want to be able to see not just blobs, we want to be able to resolve very small things using microscopes. And in a light microscope, you're generally limited to this idea of half the wavelength of light. To kind of put a cheat sheet up if you don't remember your light wavelengths, they correspond to color. So we're talking about 150 nanometers on a good day would be like a single pixel in a microscope image. Again, this is just to kind of emphasize that these things are on very small spatial scales, but light is limited and electrons are much smaller than the wavelength of light, and so we can kind of leverage those to electrons to get high resolution. So then there's the idea between scanning and transmission microscopes. It's really quick. So scanning, you might use a magnifying glass to look at the surface of something. Transmission, you want to cut something really thin and then you want to look through it. You want to see how light or electrons kind of interact with your sample as they pass through a thin section of your sample. So for example, you want to look at a fruit fly eye, you could, with a magnifying glass, you can see all the nice features of the compound eye, and you can start to maybe see some of the big pronounced hairs on the fruit fly, but you can't see the brain and you can't see the guts and you can't see the flight muscles. But if you use a compound microscope, as you see on the right, hopefully you can see those things. You can start to look at those in detail. And there's the idea between light microscopes and electron microscopes. Okay, so light microscopes on the left there, they can range in price, and you can get kind of inexpensive light microscopes, or you can get very, very expensive light microscopes you can see on the bottom. But electron microscopes, everything has to exist inside a very high vacuum, almost the vacuum of space. And so living things generally can't survive that. And so things that we put into the electron microscopes are almost always very, very dead and also dehydrated so that they don't turn to raisins. And often heavily processed. So you can't really examine living things in the electron microscope, but you can see a much higher resolution. So here's kind of an example of like how you can use light and electron microscopes complementary ways. This is a tardigrade or a water bear that you can find in moss. You see kind of crawling around in a light microscope. You can't see all the fine features like its mouth that you might see in the bottom panel here. You can see its mouth is kind of this interesting circle shape, and here you see some of their claws in very high detail. So this is kind of how they can be complementary, but you can get different kinds of signal. So here's some more kind of data that we kind of look at in our work a little bit more. Here, using a light microscope, you can see kind of a tree in the forest of the brain, for example, and we can go into that a little bit more after, maybe in the Q&A period, if anybody's interested in talking about this organization of the brain a little bit more. But you can see like this is in an awake mouse, you can see like labeled neurons kind of pulsating, and that's how they exist in life. Here you can see in a cultured cell, you can see mitochondria. And this is from Kara Shivan that works with core and Dr. Manor. And you can see the kind of behavior of mitochondria inside a single cell. Here you can see in a behaving zebrafish, you can watch individual neurons firing as it's kind of embedded in agar, it's not moving much. So again, in light microscopes, you can get this nice resolution. You can get high resolution, you can see lots of things, you can look at dynamic behaviors, but you can't get the same resolution as inside of an electron microscope. So here we're looking at kind of what Uri showed you before, those synapses. We can start to look at those in very high detail using an electron microscope. And we can also extend those ideas to 3D and start to look at these structures that might look similar to what we see by fluorescence microscopy, but we can really investigate them in much more detail and at a different kind, and ask different kinds of questions. So I'll get back to that video later in the presentation, but hopefully that's a good kind of overview of different use cases for these different kinds of microscopes. Huh. Huh. Okay, sorry, I seem to have hit a technical challenge here. Okay, can you all still see, can you all still hear me here?
Yeah, we can hear you. And I can see your, see like your whole PowerPoint window.
Oh, perfect. Okay, sorry, yeah, just kind of froze out on me there. So now we can just jump right into the technical workflow. So the idea is if somebody comes to the core with a sample they want to look at with using 3D electron microscopy, this is kind of what the workflow might look like. We receive the samples, we fix them with aldehydes so that they don't rot or anything like that. So they're very preserved with very high structural integrity. Then we can stain them with heavy metals. We can embed them in plastic. We can section them with diamond knives. We can image them with these electron microscopes under high vacuum. And then we can, the rest of this will become a little bit more, make a little bit more sense as we move through the workflow here. So briefly starting with the dissection idea, this is Leo Andrade, the electron microscopy master that works with the core. And here he's dissecting the inner ear, kind of what Uri was showing in his presentation. This is what the first step looks like. So if we were to get an inner ear from a collaborator, we have to go through, or Leo has to go ahead and dissect away all of the fat and bone and things that aren't the tissue that we want to look at. This can take an hour or for a single ear to dissect. And here you're starting to see him to pull out the spiral cochlea there. And then he has to take off the tectoral membrane, which is what those hair cells embed themselves in that Uri was talking about. And so this is a very challenging preparation, all under a microscope with very steady hands. And then after you dissect the sample, you fix it with aldehydes, and you stain with heavy metals like osmium and uranium that can interact differently with the electron beam, and they bind to things like the cell membrane and to different proteins. So we can provide stain to provide gain for the features that we want to look at in our sample. And then we can further process it by replacing all the water with ethanol, and then replacing all the ethanol with an epoxy resin, curing that in an oven, and then we can prepare that. And this is what that cochlea might look like. This piece of that spiral looks like inside of a piece of epoxy resin. So this is a little, after it's stained with metals, it kind of turns black. It's this little black strip. And so this is what that can look like under a scanning electron microscope. This is what Uri showed in his presentation. But if we want to look at it, if we want to look at the inside of these cells, you can't see that with the scanning electron microscope in quite the same way. So what we can do is we can cut serial sections. So on the left here, you can see, this is called an ultramicrotome, which is kind of like a really, really fancy deli slicer. And what it does, we put a diamond knife on it. So this is a perfect diamond edge, about 6 millimeters wide. And we have the sample kind of advancing very slowly, about 70 nanometers every time you want to take a slice. And we have it's called a boat, but we have the knife filled with water. So that as the sections come off of the knife edge, they float out on water and they create this perfect ribbon. And then we can collect those ribbons on silicon chips that you can see here. Some of them are more perfect than others. And then we can put these chips inside of the electron microscope. And so once we look at them inside of the electron microscope, we can start to see some of the detail. And hopefully this starts to look familiar based on the video that Uri showed in his presentation as the inner ear cochlea in cross section, right? So here we can see that there's axons that will go to the brain to transmit the signal from the hair cells up here in this box. So the hair cells will synapse with neurons that project from the brain. They'll go all the way back to the brain and transmit that sound signal to the brain so that we can interpret it and make sense of it. And we can see here blood vessels with blood cells in them that are supplying energy. You can see the bone here stained black. And we can see the myelinated axons. This is almost like you have if you were to have wires, you always keep wires insulated because you don't want to lose signal as they travel through your house or through whatever project you're making. So myelin acts as insulation in the body. So you can have long range projections of neurons and they don't lose signal as they move sometimes centimeters or even all the way down your leg. There's just one neuron called the sciatic nerve that can be up to 1 meter long. And so myelin is really important in that case to make sure that you can feel your toes well and you don't get chronic pain and things like that. So, but if we look at these hair cells a little bit more closely, the cells that kind of transmit the sound, we can kind of get a sense of the difference between what we can see with the scanning electron microscope versus with the transmission electron microscope or by serial sectioning of the electron microscope. And the idea is we can see the inside. So here, if we look in this square, you can see that there's a nucleus here. This is where the DNA is. And we can highlight all of the mitochondria here. And we can look at one in particular, and this is where all the energy is made for this, or well, where energy is made and kind of converted into usable energy for the cell. And so we can, and as Uri demonstrated in his presentation, the mitochondria can respond to injury and they can respond to all kinds of conditions. And so understanding the distribution of mitochondria in a cell might be a really important question to ask. And so how do we start to get 3D information from these serial sections? Well, we can stack them all up and we can align those sections. And once we do that, we can wind up with something like a block here of tissue. And if we segment, kind of color in the lines for each mitochondrion and for the nucleus, we can start to get some 3D representations of these individual components of these cell types, right? And that's how we can start to build these 3D models. And then from there, we can go towards analysis. And I won't go too much into analysis today. I'm going to kind of stick more to visualizations. This is from Tammy Pham, who's a medical student who really was a wonderful collaborator, and she kind of did a lot of this tracing work and also did this visualization, which I think is just wonderful. So, and she's kind of leading the analysis side of this project in terms of trying to figure out how mitochondria are distributed differently before and after noise. So this is kind of how we can start to study that. You can imagine you can just take a distance from the nucleus for each mitochondrion, the center of each mitochondrion to the center of the nucleus, and you can look at the different distributions of distances based on before and after noise and start to understand if that's really a salient response to that injury there. So for the next part of the talk, I'd like to highlight some of the work that that myself and other collaborators in the core, or other my peers in the core, Leo Andrade, and Linjing Fang, and Arlo Sheridan, have been working on over the last few years, and kind of projects that we're working on day to day here. So here's a project that we worked on in terms of where we were looking at the brain, and just a refresher on how the brain's organized. Brain cells are kind of organized like trees, where you have branches that receive signal, kind of like how leaves receive light, or you have leaves on branches, how that receive light, and similarly you have these dendritic spines with synapses on dendrites that receive signal in the form of neurotransmitter, kind of how Uri explained earlier. And here you can see that these synapses can happen on dendritic spines, which is what we're looking at in this right now in the video here. You can get a sense that the blue is the spine here, and it kind of plugs on to this axon here. And here we can also see that sometimes synapses happen on the shaft of the dendrite, where here we see that there's some mitochondrion and a bunch of synaptic vesicles right opposed to this dendrite here that also has some mitochondria inside of it. And the relationship between shaft synapses and spine synapses is really important for information processing in the brain. And so that's and this is really one of the most effective ways to study these kinds of phenomena. So, but it's very time consuming to trace out these processes by hand. So kind of what Uri hinted at, and what Arlo Sheridan is really an expert at in the core is how do we take those human training data sets and extend those just to massive data sets, for example, an entire fruit fly brain. And so here's an example of how what the training output can look like. So here is Arlo's program filling in a single dendrite, how I might try to do it, but it might take me 100 hours to do this single dendrite really precisely, where it might take Arlo's algorithm on a really, really powerful computer, it's mere minutes. And so you can imagine how this can scale to a really dense volume. If you want to segment all of the features in here, it would take a very hard to quantify amount of time for a human to segment this whole volume in such a saturated fashion. In fact, one of the leaders in the field, Kristen Harris, has been building a saturated volume by hand for almost 20 years, and she's still kind of updating it with detail to this day. But you can see that Arlo's algorithm can do this in, again, matter of minutes or hours automatically using the training data sets, and this can be extended to the whole brain, where we can determine in a fruit fly different projections from one region to another, and even calculate all the different synapses between cells from one region that project into another region. And so I encourage you to ask Arlo about some questions about how he did that in the chat. But as kind of already mentioned, it would be totally impossible. A fruit fly brain is one thing, but a mouse brain would have, or a human brain would have more information than the whole internet. And so it's not really practical to image at very high resolution. If we want to count all the vesicles in every synapse, we simply couldn't do that at this point. So an idea that what we could do instead is we could image everything at low resolution, and then image subregions at high resolution, and train a deep learning net to be able to calculate what the high resolution looks like from the low resolution. And that's exactly what Linjing did here. And that enables us to do very very effective segmentation, where we can look at the relationship between not just one cell and another cell, but inside of one cell, looking at different components of the cell, like vesicles and endoplasmic reticulum and mitochondria, and seeing how those relationships change under different conditions as well. And then changing gears completely, happy Halloween, everybody. This is another idea of what we can look at inside of a scanning electron microscope. So this is an image that Leo Andrade took of a spider. I'm sure most of you would recognize it, but maybe you didn't realize just how hairy spiders can be. And maybe you thought, like, well, you know that spiders have mandibles, you know, they have eyes, but maybe you've never realized that they had so much hair around their eyes. And maybe we could take a closer look at, you know, what that hair around their eyes looked like. And you can start to see how there's different lengths of hairs. And if we go a little bit closer, you can see that some hairs even have lots of bristles. And you can see that there's actually stomata, and this is how air exchange can happen in insects. They don't have lungs like we do, so they have little holes all over their body where they can achieve air exchange. So this is another example of kind of how the scanning electron microscope can kind of give you a different kind of insight into everyday things. And with that, I don't want to go over time. I do have a few more slides here, but I wonder if it's time for a Q&A at this point. So I'd like to thank everybody at the core and you all for your attention, and thanks so much for the opportunity to present some of our work here today.
Awesome. Yeah, we have a couple minutes for Q&A. If anyone, you're welcome to unmute yourself if you want to ask verbally, or continue to type them into the chat. I did have one quick question. Why does this, in one of the early slides, you showed and said that it needed to be in a vacuum? Why does it need to be in a vacuum?
Yeah, so an electron beam is kind of different to a light beam, in that electrons can interact with air and other kinds of particles in atmosphere. So you can't just shoot an electron beam; it'll interact with everything. You can't, and so you have to make sure that there's no particles in your chamber where you want where you're focusing your electron beam. And so you need to pump down that chamber down to almost the vacuum of space.
Awesome, thank you.
That said, there is a group in Japan that has a really interesting technique to create almost like a body armor that seems to work for some insects. You can look at them in a scanning electron microscope while they're behaving. But it's not a very popular or technique yet, because it's quite new, but it's pretty exciting.
Looks like we do have a question in the chat from George. Are quantum computer architectures influencing brain function hypotheses?
That is a great question. Maybe somebody in the chat can field that better than me. I'm not a quantum computing guy. I think that if you wanted to look at, if you're building a quantum computer and you want to make sure that you have like quality control or something. Or for all computing applications, really like quality control is done inside scanning electron microscopes, and most processors that you're using in your computers to watch this video are built in focused ion beam milling machines, which operate under vacuum, and we use those also for some of the imaging that I presented. We use focused ion beams combined with scanning electron microscopes so that we can mill away one layer and then image, and then mill away the next layer and image inside of the microscope, and that way we can also get much higher spatial resolution in Z. But that's not a scope that we have at Salk right now, but it's one that I've worked on before in Canada. So we have access to some microscopes like that. And we have collaborators that work with microscopes.
Awesome. We have a question from both Robert and Adrian. Would like to know, does your work require coding and biological degrees?
Oh, that's a great question. So I'll say my degrees are in biology. Firmly, and so Arlo can really testify to my I'm filling holes constantly, but I think every time when I've had the opportunity to speak to students in high school or middle school, generally if they're thinking about college applications and if they're interested in biology, I generally recommend they go into something like physics or engineering for an undergraduate degree, because generally speaking, I think biology from my perspective, looking backwards, it's a lot easier to just teach myself some of the biological concepts than to teach myself linear algebra or differential equations, or how to apply those in those kinds of ideas in a programming framework. That said, there are lots of great online courses through Coursera. So I taught myself Python and a bunch of other programming languages online, just in order to do the tasks that I needed to do at the time. And then over time become more or less proficient in being able to cobble together tools. But I think if I had a foundation in computer science or engineering or physics, where those kinds of kind of more technical skills are more enforced, or more like kind of part of the curriculum, I guess, that might, looking back, that would be really helpful for me right now. Yeah. Hope that answers the question.
I want to tell a story about one of my heroes in the imaging field. His name was Michael Davidson. He died of cancer a few years ago. But one of the amazing things he did, so first of all, building on what Sammy said about degrees, it's much easier. If you have a degree, it opens doors. There are certain grants you can get, certain types of jobs you can get if you have a PhD or a college degree that you can't get without. But there is one guy named Michael Davidson who decided to buck all of the conventional trends. And he, first of all, got private funding by selling microscope images. So he built his entire research operation through sales and decided he didn't care about the government, didn't want funding from any of the traditional sources. And the other thing he did that was really cool was he hired high school students and dropouts. He had a soft spot for kids who had suffered or made mistakes or something in the past that made it harder for them to get degrees and to go down traditional paths. So he would hire these high school students who his only requirement was that they were smart and motivated. And he taught them biology, how to do molecular biology, how to do microscopy. And there are a lot of examples of that with computer coding as well. These things seem esoteric because they're not part of our daily lingo. But the reality is that everything can be broken down into some simple components and anyone can learn them. And you shouldn't be daunted by whether you have a degree or not. It's within your grasp. It 100% is. So that's my answer to that question. I hope it at least provides some sort of answer.
Thank you. Looks like we have time for probably one more question. If anyone has any final questions for our presenters today. Again, you're welcome to unmute yourself if you'd like to ask verbally.
Or maybe I'll just give another plug for there's the software that I use for all the 3D stuff. I try to use all free software, And so it's all open source, free software for all the analysis, and a really cool piece of software that I've been working with for a while now is called Blender 3D. And that and there's some really wonderful tutorials. You can make full-length animated movies in it, like similar to Pixar movies. There have been a number of movies made in the last few years to kind of show off all the different kind of workflows and kind of tools that you have built into the software. And it's really one of the most exciting things that I'm working on these days, and it's totally accessible for students. And there's a really wonderful YouTube channel called Captain Disillusion, where he goes over a lot of the, he uses Blender for a lot of stuff. And he also talks a lot about a lot of these concepts like resolution and frame rate and color, in really, really cool, intuitive ways. And I think that they could be integrated into lesson plans well. And I can kind of pop those in the chat now as well. That was something I was thinking about this morning as something to plug. So maybe we'll do that at the same time here.
All right. I would like to say thank you so much to the Biophotonics core team for joining us today. Your passion was evident. It definitely came through, and you definitely sparked a lot of ideas for myself, even including it for myself, and ways that we can show our students that while science is important, understanding the tools that we use to do science is just as important. So I'd like to say thank you so very much for joining us and spending a couple hours with us. Ellen, would you like to say a final word?
Thank you very much. As I said, my wife's a teacher. Teachers had obviously a huge influence on all of us. So we're all so grateful for all of you for taking your time on a Saturday morning to, let us talk about our work. And if there's anything we can do ever to help, we can come in on a Zoom and talk to your students, or, really anything we can think of, we would love to help. And again, thank you for all that you do and all that you are.
Well, Uri said pretty much what I would like to say too, but I also wanted to thank Madison and Josh for actually keeping the educational outreach team going. It means a lot to me, to see that it's in such good hands. And this is a different type of way to interact, and I really appreciate everybody being part of it. And as we move forward, we really expect to be able to do this more so that we can invite more people. This time we are international. So we have people from Canada and even from Buenos Aires if they clicked in. So again, thank you to everybody. Education and getting students inspired is what we really, you could see, saw a comment, everybody's so passionate about their work. And that's all starts from their initial learning and being excited about learning. And that's the role that we hope we have, inspired you guys. And so thank you so much. And I'm looking forward to be part of the breakout groups and everyone joining us for that. And I'll pass it back to Madison.
So again, scientists, thank you so much. We will definitely share the recordings, and I will share the PowerPoints with all of the teachers in attendance here today. So for our fantastic teachers, we're going to be, just as Ellen mentioned, we're going to dive in just a moment to our breakout rooms. For our next activity, you will need your cell phone. So we'll take a quick 1-minute bio break. And if you could grab a cell phone so it's nearby, we'll get started to get into our breakout rooms at 11:16. All righty. So what we're going to do now that before we go into our breakout rooms is we're going to use a great virtual engagement tool called Mentee. So if you could go to the website www.mentee.com and then you'll use the code 9252676. And as it looks like a couple people have already figured it out. And what's great is your reactions will populate live and we'll be able to share that with our presenters. All right, looks like interesting, mind-blowing, epic, exciting, interesting, super interesting, sorry. I would agree. Fast-paced. Yeah, science changes very, very rapidly. I think that's something that's always inspiring and interesting to me. All right, we have 9 people joining. We'll give everyone else a little bit longer to join. Oh, love all the responses. Thank you. All right, I'll give it maybe another 30 seconds and we'll go on to the next one.
Madison, I was going to recommend if teachers are not using this in their virtual classrooms, if you're doing that, this is a fantastic tool. Maybe you can talk a little bit about some of the other things you could do with Mentee. Yes,
so Mentee, there is some other questions. So this is a word wall, and we're going to have an opportunity to kind of take a look at some other ways that you can respond to Mentee, but it's a great tool to poll your audience. There are some controls in it for profanity or certain emojis you might not want your students using. You know, they're creative, they figure out ways. So it's a really great tool for that. All right, we will go on to our next question. Oh, I love all the emojis are so fun. All right, so on your registration form, I had asked you all, what is currently your greatest challenge? And these are some of the most common responses. So I would like to know if you agree or do you also relate to these challenges, or are these less of a challenge for you? If there's one that doesn't necessarily apply to you, you should be able to skip it, but you're going to scroll, use the bar to give us a general idea of what your response. You should be able to scroll. Oh, wow. Awesome. Hands-on activities at home and in hybrid model seems to be a big challenge for many. Engagement. Ooh, virtual labs, okay. Love seeing all the variants. It's really helpful. I'm curious if I had asked you this when you had registered maybe a month ago, how you would have responded compared to how you would respond now. So I think across the board, it does seem that we're all kind of on similar pages of things that we're finding to be challenging in these current circumstances. So as we go into the breakout rooms here in just a few minutes, I think it's important to remember that we're all kind of in this together. And although we might be in different states or teaching different subjects, it could also be we're joined together by as educators in some of these common challenges. So my final question for you for the Mentee was, so this is an example of, like Donna had mentioned, a different type of tool. So it'll tell you the number of responses that you had at different parts on the scale. So it's a really great way to collect live data. It is, if you have the paid service, you can actually download the responses in an Excel, which is pretty cool. Otherwise, you'll just download them as a PDF. So when the survey responses came in, a lot of teachers said engagement was their biggest challenge. But I wanted to know is, how are you defining that term under these current circumstances? What do you mean? What does engagement mean to you? How are we saying if we're struggling with engagement, what does that mean? Students attending Zooms. Oh, interesting. Responses and work products. Okay. Actively participating in class, speaking, student interactions. I know in San Diego, students are not required to turn on their cameras. So we've definitely had the experience of teaching to black boxes so we can relate. Are they interested in the work there? Oh, so great. Answering questions, whether in the chat box or unmuting themselves during sessions. Absolutely. So I think it's really important just to kind of recognize again that we're seeing a lot of similar trends in all of our groups. So I think using the breakout time to try and identify ways where we can continue to work towards engagement and present content that is relevant in the real world beyond the classroom walls, I think is really important. And I will definitely save these and send them to all the participants in the Google folder so that way you'll have access to it. So I'm really happy to say that Education Outreach does have some programs that can be used to help hopefully engage your students in another way and hear another voice. So at this time, I'm going to pass it over to my colleague, Joshua Von Trapp, who's going to walk us through just a couple of our programs, and then we'll go through the breakout resources and head into our breakout rooms.
Awesome. Thanks, Madison. I'm going to take over screen share. All right. Could you give me a thumbs up on the Zoom box if you can see my shared screen? Awesome. Thanks, Madison. All right. As Madison mentioned, yes, we do have a lot of programs that we wanted to highlight. One program I'm particularly fond of, thanks in huge part to Ellen Potter of the same symposium name, is our Mobile Science Laboratory. Since 1996, we've crisscrossed all over San Diego County, serving around 20 schools and around 2,500 students annually. And now we just wrapped up our 4th week of our virtual MSL yesterday, but I'll share more on that later. Another program I'd like to highlight also with a long history at Salk is our March of Dimes High School Science Day. Each year, around 300 high school students are welcomed onto Salk's campus for an up-close look inside of our labs and direct interactions with scientists. We're happily exploring virtual options for 2021. And again, speaking of traditions, our Heidoff-Brody Summer Scholars. Each year, from a large applicant pool, we select 10 to 12 high school students for a very special paid internship opportunity to work directly in the labs. And it's most definitely not just washing tubes and glassware, it's so much more, and is completed with a capstone presentation by each intern. But as Madison noted, and as we all are on this virtual call, we're in a pandemic pause. But to meet that moment, of course, EO went virtual. So right now, as you're listening to these programs, think about which one might be a fit for you and which one might be a fit for maybe a teaching partner to pass along to. So our SciChats program is a program that existed pre-pandemic but has really taken off with our increase in distance learning. Though whether you're in person, in class, or in a box on Zoom, SciChats are all about connecting scientists with students. So whether it's to share their career path and latest research, or possibly a lab tour demonstrating a technique, it's a great chance for students to break their stereotype of what a scientist is and does. And just yesterday, there was a human brain on the SciChat Zoom, which completely blew all of the 8th graders' minds on the call, including mine. Now, as mentioned on our previous slide, our virtual mobile lab is in full swing. While we have a fall calendar that's fully booked, we will be opening up our winter calendar for virtual visits happening January through March, and that calendar will be opening up in November. We offer live learning with scientists and even include a hands-on DNA extraction that each student can participate in either in class or on the Zoom call with us with the supplies provided by us. Lastly, Introduction to Research Science and Communication virtual program. We piloted this 4-week online experience this past summer as a way to meet the moment when our in-person internships like Heidoff-Brody were just not possible. But elements of this Introduction to Research Science VP include strategies for reading a journal article, representing and analyzing science data, and creating and delivering a presentation for a lay audience. Feel free to reach out to us if you have any questions about those programs in the chat or later on. As well, there's a few teacher resources I'd really like to highlight, 3 especially. The first, you probably received this morning an email from Madison and should be dropping in the chat for when we head to our breakout rooms. Those resources are to be used for our teacher symposium and hopefully in the days afterwards. As well, we have lots of other resources on our website that I would like to highlight 2 more specifically from this list. The first And second are our virtual lab tours and our action science video series. If you're looking for visual content to enhance or supplement your lesson, these two are great options to explore, especially with the action science series has docs and materials that you can begin to use immediately, like on Monday. All of the info will be available for us on our website, or you can reach out to us in the chat. And then at the end of the symposium, we'll be happy to leave the lights on in case any of you want to stick around and ask us any more questions about any of these programs. On that note, I want to pass it back over to Madison, who's going to be sharing about our collaboration and breakout rooms. Madison, you there?
Sorry about that. I was just trying to grab the link for the resources folder. Would you mind dropping that in the chat for me, Josh?
Absolutely.
Thank you so much. All right, so today our activity, so when I looked at the breakout rooms and I was trying to determine how to break everyone up, I decided it probably made the most sense to assign you to breakout rooms based on your grade level and content area, because so many of you had indicated that you were interested in many different options, discussing challenges, talking about NGSS alignments, and looking at the virtual lab simulations. And what you would wanted to look at may have even changed after today's presentation. So it's really going to be a choose your own adventure. If you leave the breakout room excited, inspired, with something to share and bring back to your students, we're going to say that is successful. We do have some resources to help get you started, to serve as like a jumping off point. Josh, it looks like it had just flipped back from the screen. Thank you. So we do have some resources in the Google Resources folder. So this is what it should look like. And if you'll just go to the tab that says Resources, and I'll talk just shortly about what's in there so you know what to expect. So I did some potential NGSS alignments based on the outlines that the presenters had shared with me. So these are not to limit you. These are just jumping off points. You might totally think and take it in a different direction. I did a couple for middle school and then a couple for the different high school subjects. I did want to note that these were taken from the California state standards. So there might be different if you're teaching in a different state, but should be generally pretty similar, as these standards are national. We also found a handful of really cool virtual lab simulations. Unfortunately, because Salk's a nonprofit institute, we don't have the funds just yet to develop our own virtual lab simulations, but there's a lot of really cool resources out there, so there's no reason to reinvent the wheel. So this is just one quick example of a virtual lab simulation that we were able to find. There's also links to some of the websites that our researchers had talked about, like the Tracing the Deep Roots project and the image cell library, if you want to take a look through those as well. We finally, if you would like to spend the time discussing challenges with your group, we have a template in there with some directions on if you want to add to it as part of the live document, you're more than welcome to. Otherwise, it can just serve as a prompt for conversation. And please don't feel limited by these resources and options. If your group wants to spend the time discussing how cool the presentation was, or how different technology, or how much technology has changed since you went to college, that is perfectly okay. If you want to spend time anticipating student misconceptions, that's great too. As long as it serves your group's needs, that's great. There's not necessarily a specific action item or thing to fill out during this time, with one exception. We're going to use a tool, another really cool virtual engagement tool called a Google Jamboard in our wrap up. And I'll go through that in just a second, which is how to use a Google Jamboard, but it's basically a virtual brainstorming tool. So we're going to ask your group to add a Post-it note to the Google Jamboard to share your group's takeaways. You're welcome to add notes to other people's posts or just add your group's your own. That can be done on by a group, Salk team member or any teacher. So if you've never used Google Jamboard, it's really great way to get students collaborating together virtually. And I know that I saw that pop up into how we're defining engagement, so maybe this could work. So what we'll go through next, this is a quick screen capture of how to add to a Google Jamboard. So inside, we'll just wait for it to play. Oh, technology. So inside the teacher resources folder, Josh, you can stop the screen share and maybe we will just all go through it live. Okay, so I'm going to take over screen sharing really quick. So you're going to go to the resources folder and you're going to identify the one with the orange icon, and that's our Google Jamboard right here. Once you click into it, it'll bring you to our shared whiteboard space. And to add a Post-it note, what you'll do is add the Post-it, the sticky note, and then you're able to change the color and add a message. You can then cancel it and you're welcome to move it around. If you'd like to edit, you can click the hamburger menu and you can edit it, order it if you want to put one on top of another. If you'd like to add an arrow to address another person's comments, there is a nice little marker button that you can use. If you're not happy with your mark, you can erase it. This is a great tool. If you're an experienced Google Jamboard user, you're more than welcome to add a picture or a text box, whatever you wish. So what we're going to have now is you're going to be sent into your breakout rooms. So if you've never used a breakout room on Zoom, you're just going to click the join and it'll bring it to you. And we're going to spend about 15 minutes in our breakout rooms, and about 5-minute warning, you'll get a warning. So please just make sure to add to the Google Jamboard if you haven't done so by then. And then we'll get back together for a wrap up and end right about on time at 11:55 noon. All right.